Hello guys, so today we are going to discuss on the topic tRNA and its structure and function. So, have you ever wondered how your body reads the genetic code and actually builds protein from it? The answer lies in this tiny hardworking molecule called the tRNA. So, today we are going to dive deep into this hero of protein synthesis, the tRNA, as also called as the transfer RNA. So, hi, it's from BiotechKing, a biotechnology student passionate about making complex biology easy to understand. So, welcome to my channel. Today, we are going to discuss about tRNA, the process, and how it functions in the process of translation and why it's so essential for life as we know it. Whether you are a beginner or revising for exam, this is for you. So let's move on to the structure of tRNA. tRNA, as the name transfer RNA, has a length of approximately 70 to 90 nucleotides long and plays a crucial role in protein synthesis. So it translates genetic codons that is present in the mRNA into amino acid and thereby building proteins. So it helps in the formation of amino acid, which is the building blocks of protein. So let's move on to the structure of tRNA. So the tRNA basically has a clover leaf secondary structure, and the structure is very important as it helps it carry amino acid and recognize mRNA codons. So let's talk about different arms of tRNA and its function. First one is the acceptor arm, second anticodon arm, D loop, pseudouridine arm and variable arm. So at the ribosome, the tRNA brings the correct amino acid to build the growing protein chain. So without tRNA, the cells won't be able to turn genetic information into proteins. Like life simply doesn't function. Moving on to the function of each arm. So this is the basic structure of tRNA and the different arms of tRNA. The acceptor arm, that is 3 dash end. So it always, in every tRNA, it always ends in a CCA sequence, and this arm is the one that accept the amino acid that helps in translation. So it has an end which ends in CCA sequence where the specific amino acid is attached. Next, we are going to talk about the D loop. The name comes from dihydrouridine arm because it contains dihydrouridine. So it involves in tRNA recognition by amino acid tRNA synthetase, and it has a length of approximately 7 to 11 nucleotides. So, the amino acid tRNA synthetase is an enzyme that plays a key role in binding of amino acid. So, this activates the amino acid that helps the amino acid to bind to this end, that is the acceptor arm. Next, we are going to talk about the anticodon arm. So, it contains a triplet anticodon, and this codon is complementary to the codon of the mRNA. So, it has 7 nucleotide long, and hence it is called the anticodon loop because it pairs with the mRNA codon, and hence the name anticodon. Next, we are going to talk about the pseudouridine arm, and this arm contains the ribothymidine, pseudouridine, and cytidine molecules, and it has a length of 7 nucleotides long. So it is very important for ribosome binding. Next is the variable arm, which we can see in the above This is the variable arm that is given in this yellow kind structure. So it has variable length among tRNAs. So it can be from 3 to 21 nucleotides. So it recognition of tRNA by amino acid tRNA synthetase. So these are the 5 different arms of tRNA. Now let's move on to the function of tRNA. So tRNA serves as an adapter molecule in translation, which converts genetic code in mRNA into a specific sequence of amino acid in a polypeptide. So the amino acid carries uh, the amino acid carrier, that is each tRNA is specific to a single amino acid. So the enzyme amino acyl tRNA synthetase helps the amino acid to binds to the corresponding tRNA. So this is the equation that I have given. So tRNA with amino acid in the presence of amino acyl tRNA synthetase gives a complex or charged molecule called the amino acyl tRNA. Next is the codon recognition. The anticodon of tRNA base pairs with the complementary codon and mRNA during translation. And this mainly happens in presence of ribosome. So when we talk about amino acid tRNA synthetase, it's a key enzyme that plays a crucial role in protein synthesis by linking tRNA with corresponding amino acid. Next, we are going to talk about how tRNA is being helps in translation of the mRNA. So this is a ribosome and this is the mRNA. These are the 3 sites or compartments in a ribosome. So there are 3 major compartments: the A site, B site, and E site. And different sites has different function. So A site, also called the amino acyl site. So this site welcomes the new tRNA which is carrying a particular amino acid. So hence it is called the amino acyl tRNA. Next is the P site. After binding to the A site, the tRNA carrying the amino acid jumps to this site called the P site, also known as peptidyl site, because the tRNA, the amino acid, binds to form a polypeptide chain in this P site. And after the addition of amino acid to form the polypeptide chain, the tRNA jumps to the other site called the exit site. So the amino acid, so the tRNA that lacks or emptied amino acid is being released into the environment. And now the E site is empty. Hence, it is called the exit site. So, the ribosome shift over the mRNA by one codon, and this is called the translocation. The tRNA in the A site moves to the P site, and the empty tRNA in the P site moves to the E site where it exits. So this is the major function of tRNA and how tRNA helps in translation of mRNA to form proteins from the building blocks of amino acid. So that's all about tRNA, its structure and function. Thank you for listening to my slide. I hope you all understand. So if you want to know more about biotechnology and biotechnology related topics, you can subscribe to my channel. Thank you.